Welcome back, everyone. During this chapter, we are going to go over the chemical level of organization. In the last chapter, or chapter 1, we talked about the properties of life, the properties that were exclusive for something to be classified as life. We went over the different levels of organization, as well as a little bit into detail about homeostasis, what a receptor is, what an effector signal or afferent signal is, what an efferent signal is, as well as what an effector organ is. So this chapter kind of goes back to that chemical level of organization. So during this chapter, we're going to talk a little bit about what organic chemistry is and go over the 4 macromolecules of life. Each and every one of these macromolecules of life has a specific function to play within the human body. And those 4 macromolecules are carbohydrates, lipids, nucleic acids, and proteins. And the organic chemistry is a branch of chemistry that deals with carbon-containing compounds. So if something has carbon, it is an organic molecule. Now, organic molecules can either have affinity towards water and would be called hydrophilic, or they could repel water and would be known as hydrophobic. So hydrophilic molecules are ones that are water soluble. Hydrophobic molecules are ones that are not soluble or do not dissolve in water. An example of a hydrophilic substance would be table sugar, sucrose. So sucrose, when you put a packet of sugar into water, it dissolves. An example of a hydrophobic substance would be olive oil. So if you take a teaspoonful of olive oil and drop it into a glass of water, you can mix it all you want, but it will eventually separate into 2 separate layers. So that is because olive oil is an hydrophobic substance. So the first macromolecule that we are going to go over is carbohydrates. Carbohydrates are varied in their roles in the human body. They are organic molecules and can be very small or very large in size. So they have a wide variety of sizes. They're relatively simple. So carbohydrates contain carbons, hydrogens, and oxygens. Now, different examples of carbohydrates. Carbohydrates can be broken down into simple carbohydrates or complex carbohydrates. An example of simple carbohydrate would be glucose, or blood sugar, or fructose, or fruit sugar. A complex carbohydrate would be something like starch, or cellulose. The functions of carbohydrates are varied. Most of the time, they are required for generation of biological energy, or ATP, which is adenosine triphosphate. So when you ingest carbohydrates, your digestive system is going to convert it to glucose or blood sugar. Glucose or blood sugar goes to every cell in your body and participates in oxidative metabolism. So when I talk about oxidative metabolism, I'm talking glycolysis, Krebs cycle, and electron transport chain in order to eventually produce ATP. Carbohydrates can also be structural compounds or long-term energy reserves. In plants, for example, they are structural in terms that the cell wall of plant cells is made up of cellulose, which is a complex carbohydrate. In human beings, a long-term carbohydrate reserve is glycogen, which is an energy storage system or energy storage molecule in our muscle fibers. When we talk about the chemistry of carbohydrates, like I said before, they consist of carbons, hydrogens, and oxygens. Now carefully look at the ratio of carbons to hydrogens to oxygens. For every 1 carbon, you are going to have 2 hydrogens and 1 oxygen. So one of the simplest carbohydrates is glucose or blood sugar. Now in blood sugar, there are 6 carbon molecules, 12 hydrogen molecules and 6 oxygen molecules. So C6H12O6. Now, carbohydrates based on size can be categorized into monosaccharides, disaccharides, or polysaccharides. Now, monosaccharides are the monomers of carbohydrates, meaning that they are the building blocks for bigger carbohydrates. So, simplest monosaccharide formula is C6H12O6. Now, glucose is a monosaccharide, fructose is a monosaccharide as well. So if you look at the pictures or structures of glucose versus fructose, you will notice that they both have the same chemical formula. However, what they differ in is their structures. Disaccharides are two monosaccharides that combine to form a glycosidic bond right here. So right here in the circle is a glycosidic bond. So when two molecules, you have a monomer that combines with another monomer, there has to be some molecule that is kicked out in order to make that bond. And that kicked out molecule is water. So removal of water is dehydration. So the process that is taking place when you have two monomers combining to form a disaccharide is called dehydration synthesis. So the example here in this picture is sucrose, commonly referred to as table sugar. The formula for this is C12H22O11. And you will notice that this is because a water molecule has been expelled in the process of forming that chemical bond. Carbohydrates that have more than 3 monomers are referred to as polysaccharides. So polysaccharides can be either 3 monosaccharides or they could be hundreds and thousands of monosaccharides. But the formula remains the same. So you will still notice that the carbon to hydrogen to oxygen ratio is still 1 to 2 to 1. Again, that is 1 carbon to 2 hydrogens to 1 oxygen. Now, examples of complex polysaccharides are glycogen, which is our carbohydrate reserve in our muscle cells, or starch, which is the storage of plant-based foods. The next macromolecule of life that we are going to go over is lipids. Lipids are hydrophobic substances, meaning that they are not soluble in water. Some examples in the human body are fatty acids, triglycerides, and steroids. A lot of our hormones are fat soluble or water insoluble. Lipids are also composed of carbons, hydrogens, and oxygens. However, the amount is usually a little bit lesser than carbohydrates. It has more hydrogens and more covalent bonds. Lipids serve quite a bit of purpose in the human body. One, they are insulating. So whatever adipose tissue is in the human body is example of lipids. Cholesterol is a lipid. Our plasma membranes have lipid molecules in them. Some of the most common lipids in our bodies are triglycerides. So each lipid molecule has a glycerol backbone. So this circle right here is glycerol. So every lipid molecule has a glycerol backbone. If it has one fatty acid chain attached to it, it would be referred to as a monoglyceride. If it has 2 fatty acid chains, it would be a diglyceride. And a glycerol with 3 fatty acids, as you can see here in this image, so this is glycerol and these are the 3 fatty acids, this would be a triglyceride. So the job or the goal of triglycerides in the human body is to provide padding or insulation to our visceral organs. However, conversely, the triglyceride storage in the body is unlimited, meaning that if there's more consumed than used, it is going to be stored in your body in the form of adipose tissue. Now, in the plasma membrane, so here is an example, an illustration of the plasma membrane. So the plasma membrane is what is the boundary of every cell. Now, these lipids within the plasma membrane are phospholipids, and they are split into two parts. You have the hydrophilic heads, so the blue beads here are the hydrophilic heads, which are oriented towards the outside, and the tails that you see which are oriented towards the inside, are hydrophobic. So cholesterol is stored within the hydrophobic tail region of the plasma membrane. Some other lipids in the human body are steroids. Now, steroid is also a hydrophobic molecule. It is a type of a lipid. Examples of natural steroids in the body are sex hormones. For example, estrogen, which is considered the female sex hormone, and testosterone, which is the male sex hormone. So these hormones are hydrophobic. Adrenal hormones. So we have, sitting on top of each of our kidneys is an adrenal gland, which determines how our bodies react to stressful situations versus calm situations. And the hormone that the adrenal gland secretes is cortisol. Bile salts. Bile salts are produced by the liver and are also an example of steroids. And vitamin D. Our bodies are naturally capable of absorbing the radiation, the UV radiation from sunlight, and making vitamin D. So when we go over the integumentary system, we are going to talk about vitamin D and how our body produces this vitamin. The third macromolecule group is nucleic acids. Now, these are molecules of information storage. So these are molecules that store our genetic information. And there are two types of nucleic acids. DNA, which is deoxyribose nucleic acid, or RNA, which is ribose nucleic acid. The monomer, the building block of nucleic acids, is called nucleotides. Now, each nucleotide has a few different components. One, it has a nitrogenous base. It's called a nitrogenous base, and this varies from nucleotide to nucleotide. So this is what differs from one nucleotide to the other. So this blue molecule right here in the red circle is the nitrogenous base. Now, nitrogenous bases are of 5 types, and I will talk about this in just a little bit. In the center, or the core of a nucleotide, is a sugar, which is usually a monosaccharide. So in DNA, it is deoxyribose. In RNA, it is the ribose sugar. On the other side of the 5-carbon sugar, you have a phosphate group. So attached to the first carbon is the nitrogenous base, and attached to the 4th carbon is, attached to the 2nd carbon is the nitrogenous base, and attached to the 5th carbon is the phosphate group. My bad, I just spaced out there for a minute. So, I apologize. Now, nucleic acids are important as carrier molecules as well. So they're molecules that carry stuff from point A to point B. And biological energy, which is ATP, is a nucleotide as well. So what ATP is composed of is the ribose sugar right here. What I'm circling it is the ribose sugar. To one side, you have adenine, because that is the nitrogenous base. So adenine plus ribose is adenosine. So ATP being adenosine triphosphate, you know that it has the ribose sugar, it has the adenine, which is a nitrogenous base, and it has 1, 2, 3 phosphate groups. Other examples of nucleotides is the cyclic AMP and the cyclic GMP. So cyclic AMP stands for adenosine monophosphate, and cyclic GMP stands for cyclic guanosine monophosphate. So these are responsible for communication, so cellular pathways. So these are responsible for cell signaling. When we talk about nucleic acids as a whole, DNA is responsible for storage of our genetic information. RNA, or ribose nucleic acids, is responsible for gene expression and protein synthesis. So going back here, actually I'm going to go back here, talk about all of the 4 nitrogenous bases of DNA as well as RNA. So over here, in DNA you have adenine, you have cytosine, you have guanine, and you have thymine. And according to Chargaff's rule, or Chargaff's law, an adenine can only bind to a thymine. A cytosine can only bind to guanine. You can never have an adenine bind to a guanine or an adenine bind to a cytosine. Always, adenine binds with a thymine. Thymine can only bind with an adenine. And you have 2 hydrogen bonds between an A and a T. Between a guanine and a cytosine, you have 3 hydrogen bonds. In RNA, you have adenine, you have cytosine, as well as guanine. But what is not present in RNA is thymine. Instead of thymine, there is another nitrogenous base called uracil. So going back, so we've talked about what organic molecules are, we've talked about carbohydrates, we've talked about lipids, as well as nucleic acids. So the last of the 4 macromolecule groups is proteins. Extremely, extremely important within the human body. 1, every enzyme in the body is a protein. Proteins can be transport molecules as well. So if you think about the plasma membrane, we have transmembrane proteins and we have surface proteins which serve as markers. All enzymes, so anything that helps you digest, digestive enzymes, one of them is sucrase, which helps break down table sugar that you consume to blood sugar. So that is an example of a protein. We have, in our muscular system, we have contractile proteins as well. Examples of those are actin and myosin, which I will talk about more when we get to the muscles unit. So they are hydrophilic substances, and the monomers of proteins are amino acids. There are 20 amino acids in total. However, if you look at each and every one of those 20 amino acids, they have a few things in common. So let's go over what the things in common are. So every single amino acid has an amino group, which is the NH2 group right here that I'm circling in the blue. Every amino acid is going to have that central carbon core right here. And every amino acid is going to have a carboxyl group, which is the COOH group right here. So that's common to each and every one of the 20 amino acids. What is different is what is referred to as the functional group, or the radical, or the R group. So that is what differs from one amino acid to another amino acid. So that functional group gives that protein its specific properties. For example, you're seeing two amino acids here. On the left is glycine. The functional group for glycine is a hydrogen atom. The functional group for cysteine is a little bit more complicated, and it is a sulfur-containing amino acid. When one amino acid combines with another amino acid, dehydration synthesis occurs, and that bond that forms between the two amino acids is referred to as a peptide bond. A dipeptide is a chain of two amino acids. A polypeptide contains several amino acids. Now, proteins are created in our cells by the ribosomes and our rough endoplasmic reticulum. So the protein structure is its final shape. And we're going to go over what the 4 levels of protein folding is. So right on the bottom we have the primary structure. So the primary structure is what is produced in the rough endoplasmic reticulum, or wherever the ribosomes are, basically. So it's a shapeless, two-dimensional amino acid sequence. Now from here, it goes to the Golgi apparatus, and it folds into one of two forms. So right here, in the red circle, you have a shapeless chain of amino acids. That is its primary structure. So this chain doesn't quite have a shape. It's just a chain of amino acids. In the next level, this shapeless chain can assume one of two forms. One, it can become a single helix, which is referred to as the alpha helix. And this shape is because of the way the hydrogen bonds are between the different amino acid molecules. Or it can become a zigzag beta pleated sheet. So it can become either an alpha helix or it can become a beta pleated sheet. Now here is where the tertiary level of protein folding comes into the picture. You can have an alpha helix bind with another alpha helix, a beta pleated sheet bind with another beta pleated sheet, or you could have a combo of a helix and a sheet. So at this point you have a three-dimensional protein, and that is referred to as its tertiary structure. Now you can have different tertiary structures, so different three-dimensional blobs, if I must, come together to find a bigger three-dimensional chunk. And that bigger three-dimensional chunk is called its quaternary structure. So you have 4 levels of protein folding. In the primary level, you have a shapeless chain of amino acids. This can assume one of two forms. It can turn into an alpha helix or a beta pleated sheet. And these shapes are due to the hydrogen bonds. In the third level, or the tertiary level, you can have a alpha helix and an alpha helix, or a sheet and a sheet, or a combination of both, and it becomes a bigger three-dimensional chunk. Finally, in the quaternary structure, you have 2 or more three-dimensional structures that come together to find an even bigger three-dimensional structure. Proteins, let's go over the functions of proteins. Proteins are extremely important. One, they are molecules that give you a tough structural strength. Collagen. Collagen is a protein that is present in our skin and integumentary system. So it's part of connective tissue. We have collagen in our skin, which gives us that tautness of skin. So when we lack collagen, people's skin starts to droop and cause wrinkles. Another structural protein is keratin, which is a protein in our nails and our hair. So if you look at a lot of hair products that claim to repair your hair after chemical treatments and such, they usually have some kind of keratin in them, a hydrolyzed form of keratin. A lot of anti-aging products will have collagen in them. Our bodies naturally produce collagen and keratin. However, as people age, the production of both of these proteins goes down. Proteins function in communication. So proteins are cell receptors, they are markers, hormones. Some hormones can have proteins in them as well. Membrane transport. So we're going to talk a lot more about membrane transport in our next chapter. So when I say membrane transport, I'm talking about forms of transmembrane proteins. So we have different forms of transmembrane proteins that selectively allow stuff to go into or outside of the plasma membrane. Proteins are enzymes, and they are catalysts. So for example, one enzyme in our digestive system is lactase. Lactase is an enzyme that helps us break down the sugar which is naturally present in milk called lactose. So people with lactose intolerance have unpleasant gastrointestinal symptoms if they do consume lactose. And that is because the catalyst, or the enzyme, lactase, that breaks the sugar down is not present in their digestive system. Recognition and protection. Our immune system is composed of antibodies. So when we talk about the immune system, we'll talk more about this, but antibodies are molecules, protein molecules that our body produces as a result of an infection. So the infective or any foreign material that is going to initiate an immune response or going to tell your immune system to get into action is called an antigen. So for example, an antigen could be a bacterial particle or a viral particle, or it could be a dust mite. It could be a particle of dust. So antigen is anything that elicits or produces an immune response. And that immune response is generation of antibodies. So antibodies are what our bodies produce when they come across something that they do not like or they have to get rid of. Movement. Proteins help in movement. Now when you think about the movement of the human body as a whole, you're thinking about the skeletal system and the muscular system. So when you think about muscles, there are contractile or structural proteins in muscles. Two of the most important ones being actin and myosin. So actin and myosin are proteins in muscles that help the muscle contract and relax, so thereby helps in movement. Now you have protein molecules that are part of cell junctions. Now a cell can't exist by itself. So you have to have different types of cells combine to form tissues, and they don't exist solitary. So you have to have one cell in a tissue, you have to have one cell, most tissues, you have to have one cell bind to another cell. And those molecules that actually bind two cells together are proteins as well. Examples of this is cadherin and integrin. So these are two examples of proteins that kind of help seal or bind two cells together. Now enzymes are biological catalysts, so meaning that they lower the activation energy. As a living organism, you want to conserve energy, not use up more energy. The lesser energy you use, the more smooth the process is on you. So enzymes, examples of enzymes are lactase, sucrase, lots of different ones. They're reusable, meaning that our body synthesizes these enzymes and the enzyme can be reused. However, proteins and enzymes, because they are proteins, can be denatured. Now when an enzyme is denatured, its tertiary structure is broken down, its quaternary structure is broken down, as well as its secondary structure. The one that remains unchanged, however, is its primary structure. Now, within the human body, the optimum temperature for these enzymes to work, so the optimum temperature is the temperature at which the enzymes work the best, is normal human body temperature, which is 98.6 degrees F. Now, when you have a fever, some of these enzymes may not work right. When you have hypothermia, these enzymes might not work right either. So optimum temperature is the normal human body temperature. Now, when you have an enzyme that is going to help you hydrolyze something, so look at the term hydrolysis. So I'm going to explain this image right here. So this image is sucrose, or not fruit sugar, but table sugar. Let me pick another color here. Let's do red. Okay. So this is sucrose. It is a disaccharide. Now you need to, in order for sucrose to be broken down into glucose and fructose, you have to have an enzyme or a catalyst. And that catalyst is the enzyme sucrase. So this blue blob-like thing right here is the sucrase enzyme, or the sucrase molecule. So on the sucrase, on the surface of the enzyme, you are going to have a region where your substrate is going to come and attach. So the site on the enzyme wherein the substrate is going to come and bind is called the active site. So active site is like a antenna. So this antenna is going to grab whatever is coming at it. In this case, it is sucrose. Now over here, on sucrose, you have a region that is going to fit in, so it's a lock and key mechanism. So sucrase can only break down sucrose. Lactase can only break down lactose. So the surface on sucrose is called the binding site. So please don't get confused between what the active site is and the binding site is. So the active site is present on the enzyme, the binding site is present on the substrate. So over here in image 2, you will see that the sucrose has bound to the enzyme sucrase. So this is where hydrolysis is taking place. A water molecule is going to come into the picture. It is going to lyse. One of the components, which is a hydrogen ion, is going to one of the monosaccharides. The hydroxide ion is going to go to the other monosaccharide. And both these monosaccharides are going to go their separate ways. So this is what an enzyme does. So this is an enzyme substrate reaction.